Welcome back to the 7th class in the second week. So, today before we proceed further, let's do a recap of what we decided at the beginning of the previous week. So, the whole idea was the first week I will be talking about the challenges of the biosecurity, which is the biowarfare and the bioterrorism, and the need for continuous research in designing advanced biosensors. And from here, we decided the first week we will cover these topics: agricultural security, which we have done, livestock production, human health, pandemics, and bioterrorism, what we are continuing. And the week 2, the plan was some of the spillover of the bioterrorism, and then we will talk about these 3 different kind of case studies, or rather these 4 different kind of case studies, 3 of which belongs to microbial origin of sensors, and the 4th one is a straightforward molecule insulin, which is involved in diabetes. So, the reason why I prefer to have a spillover of bioterrorism into the week 2 is to give you a comprehensive understanding why these specific topics have been selected and what are the impact of it, because it is not that we have randomly picked up certain topics. It has been picked up for a reason to increase the awareness locally, nationally, and globally. And overall, the next century will be the century of biosecurity. So, with this kind of on the backdrop of it, we will continue where we stopped in the previous class. We talked about the bioweapon race in USA, UK, and USSR. So, just to start where we left in the last class, the end of the 6th class, in the early 1940s and latter, the USA and UK and USSR researched biological weapons. Actually, as a matter of fact, in USSR, it all started in 1920 as per the records which are coming up. As a result, US biological weapon development program at Fort Detrick, Maryland, and other places, a huge arsenal was produced comprising of aerial bombs, artillery ammunition, warhead containing anthrax, botulinum toxin, tolerance, and so on and so forth. Only in the time of President Nixon, specifically in 1971 to 73, did the process of disarmament and destruction of biological weapon begin, and the Fort Detrick, now the US Army Medical Research Institute of Infectious Disease, was switched to research for defensive nature. But parallelly, there were other research which were going on across the world. So, we will pick it up from where we left. If we look at the use of bioweapons in USA and Japan, you will see the first evidence came in 1984, where a bioterrorist attack was reported in Dallas, in the city of Oregon State, US, carried out by the ashram by Osho Rajneesh. It has been said, or it has been documented, that Salmonella typhimurium was used in salad bars and infected 751 people, of whom 45 were hospitalized. This was in the recent past, I must say in the last 50 years if you look at it. This is the first instance, and this is all after the convention which was passed in 1972. I haven't talked about the convention yet because I wanted to give you some of the instances. And if you look at 1993 in Tokyo, this is even much more closer. The Japanese Om Shinrikyo, or the Supreme Truth. So, this is a sect which carried out an unsuccessful attack of anthrax and botulinum toxin. In retrospective investigation, it was shown that the failure was due to the use of vaccine strain of anthraxis not containing the plasmid PXO2. So, it is very clear after the discovery or understanding of the DNA structure, and with the advent of polymerized chain reaction PCR, commonly known in biotechnology, the race for biological weapon development or engineering biological weapon got a tremendous momentum and boost because you could produce the specific nucleic acid at a very rapid pace, okay? And in the presence of which is a condition of full of virulence. So, the sect tried to obtain a strain of Ebola, and we will talk about that later, hemorrhagic fever virus from Zaire in 1992. So, you look at it 1984, 1993 and now comes 2001. 2001 attack is, now we are into this century, was a very serious attack. I still remember that was my second year I was in US. So, it started in Florida state of US. What was done was the group which carried out this attack, they put the pathogen in the envelopes. So, if you open the envelope like something like this, you are opening the envelope, you breathe it. It was a very innovative design of weapon, and you can post it across the world because no one is detecting your postal mails. Okay. So, in 2001, the postal attack with anthrax spores in envelopes were carried out in the United States, which evoked a shocked reaction around the world. The attack led 22 people falling ill with anthrax, of whom 5 died in the pulmonary form, and more than 30,000 people were treated by antibiotic therapy. The reason why it affected heavily, as I was mentioning in the last class, no one. So, if I have a strain, I conserve the strain and I do not produce vaccines or antidotes and maintain a stockpile. Just like you do not maintain a stockpile of the pathogenic virus, you do not do a stockpile of the antidote. So, whenever such attack happens, you started again pulling it out from your kind of inventory and reproduce it. So, that takes time, and during that time you incur human animal loss. So, this in the recent time 2001 is kind of one of the very gory incident of use of bioweapon, and which created tremendous amount of fear among people, and of course, it disturbed the stock market very badly. From here we move to the Soviet bioweapon program. Now, in terms of Soviet Union or USSR, former USSR, the bioweapon program remained in dark secrets of the communist regime. So, very little really was known, but it has been said that they were working on it since 1920. So, there is a book, people are interested, you can always buy this book, The Soviet Biological Weapon Program, a history by Milton Littenberg primarily and the Raymond Zelenskas. So, this book made wise attempt to visit from the historical perspective to the present day of Russian biowarfare agent development. And there are some evidences which I will show you, which says that there was a exceptionally great program they were running, but undercover. So, one of the very first example is this. So, what you see in the green is a map, and what you see out here is a transmission electron and a, so the next one is the transmission, first one is the light microscopic picture of anthrax. So, Russian bioweapon development program was working tirelessly on developing anthrax as a bioweapon, and there was a leakage which happened in one of the villages where this particular facility was there in Silverdovsk in USSR, former USSR in 1979. Okay. And what happened is that led to the spread, and 66 people died, and this is the kind of radius where it is spread. So, all these what you see, this is how the spread happened. So, this is one evidence. It was a leak, it was, you can say, some form of negligence which led to the outbreak of anthrax in those villages. So, you see, when you run such a program, the amount of security you need, And securing such labs is extremely challenging. It's even more like you are safeguarding a nuclear arsenal. Second thing, what has been highlighted, how it has come to the notice that actually they ran a very nice program was, if you look at the publication, this is a chart of publication by two authors who were part of the major Vector Institute for Viral Research in Moscow. So you'll see their publication track hit a record number during these years, 1970s to 1980s. And same here, another spike. Interestingly, these are the two unique spikes which happened. And of course, there is a little bit here. Interestingly, if you look at it, after 1996, there is a fell down. Now, it has a very interesting reason, because it was this time when Glasnost and Perestroika led to the disintegration of former USSR. So, the concerted effort of bioweapon program development up to that time could be indirectly inferred, but post that and up to the time now, for last, you can say around 24 years of from 2000 to 2024, and prior to that, during this Putin's era, we have no idea what kind of program they are running. It is very tough to believe that they are not running, and they are at continuous war with Ukraine for last several months, those of you who are following the global news. But again, we have no idea. What all we know is from indirect sources. There is another evidence, this is on plants. So if you look at it, the Hmong refugees of Laos who collaborated with the American armed forces during Vietnam War, and I'll come to this one, this is another catastrophe, accused Soviet Union, former USSR, of attacking them with biological and chemical weapon. However, the alleged toxin warfare agent known as yellow rain matches perfectly the yellow spot. Here is that yellow spot we are talking about of the bee feces. Those of you have seen the honeybees, when the honeybees excrete on the leaves, they leave a yellow spot. Sometimes they do on your, like if there's a white shirt, sometimes I see that, there will be yellow spots all over it. It's exactly something like that. So it's extremely difficult to detect unless you have a very good sensor. You pick up a sample and just put it in the sensor. Now you're realizing why we need to understand these sensors and how generations to come have to be thorough with sensors. They should have a database. You just like you put your stuff and you should be able to say this is the possible kind of toxins we are talking about. While I was talking to you about Vietnam War, which just in the previous slide, during Vietnam War 1962 to 75, there is another thing worth highlighting. The use of chemical herbicides, especially Agent Orange followed by Agent White and similar such compounds, to wipe away the mangrove forest. So if you look at the slide, 62 to 75, both the US and the Republic of Vietnam militaries used several herbicides for tactical purposes, specifically defoliate areas to reduce cover of the enemy forces, because the Vietnamese were very good guerrilla force, visibility on the perimeters of the military installations, and for a short time to kill enemy crops. Both the US Air Force and the US Chemical Corporation purchased herbicides and used them in Vietnam. Different tactical herbicides were used in different times during that long war of almost 15 years. So when we talk about how biodiversity is being affected, here is a classic case where biosecurity of that region was devastated by a chemical agent, and which was man-made. It was not something natural. It was used as a warfare agent. So you look at it that not only you have threats from microbes, you have threat from all these different kind of chemicals which are influencing your biological resources. So, talking about this, and there is another interesting part. These were shipped all the way from US to Vietnam. Those sailors who were exposed to these kind of chemicals suffered from wide range of maladies like cancer and other respiratory disorders and neurological disorders and so on and so forth. So, while we win wars, we incur some extraordinary loss in terms of human health, and that eventually cost in a big way. If you look at the kind of molecules which were used, so this is that orange, which is 2,4,5-T and 2,4-dichlorophenoxyacetic acid and 2,4-D. So, these were put. So, these are the major used in the Operation Ranch Hand. Ranch Hand is basically, it's an aircraft, C-123 aircraft, which had herbicide spray apparatus with nozzles that produce droplets measuring 320 to 350 with rapid settling velocity. So, if you look at it, today we are talking about UAVs, and this is 1970s. They are using aircraft for the biological warfare, and these are the code names: green, pink, purple, blue. These are the most difficult one, orange and white, and these chemicals 2,4,5-T and 2,4-D. Now later, somewhere around 1980s, Environmental Protection Agency EPA of United States, they banned on the production of these chemicals because they are extremely carcinogenic and they did a massive damage not only to the biogeographic region of Vietnam, but simultaneously those US sailors, those army personnel, those people who were party to the war, they all suffered. And in a long run, the population who were native to that place too suffered. And these are some of those events which continuously teaching us that we really need to be extremely careful before using any of these agents for whatsoever purpose we have, unless otherwise it is absolutely unavoidable. We may think twice. So, Biological Weapon and Toxin Convention came in 1972. Mind it, these wars and all these things were happening at the same time. Formerly known as the Convention of Prohibition of Development, Production and Stockpiling of Bacteriological, Biological and Toxin Weapon on their destruction. This was part of disarmament in the Geneva, Switzerland. It opened for signature on 10th April 1972 and entered into force on 26th March 1975. The BWC supplements the 1925 Geneva Protocol, which had prohibitive use of biological weapons. And if you look at the detailed convention, then there are some interesting points. These are the articles which are involved: undertaking never under any circumstances to develop, produce, stockpile, acquire or retain biological weapons, undertaking to destroy biological weapons or divert them for peaceful purpose, undertaking not to transfer or any way assist or encourage or induce anyone to manufacture or otherwise acquire biological weapons, requirement to take any national measure necessary to prohibit and prevent the development, production and stockpiling and acquisition and retention of biological weapons within the state territory and under the jurisdiction and under its control, and so on and so forth. If you read through it, you will realize laws were there in place, but then laws are made by human. How much are we following them? It's extremely important in the current state where more and more molecular biology techniques are advancing and more disturbances are spreading. More and more people have access through internet of technologies. The biosecurity protocols will have to go hand in hand if the problems are increasing in arithmetic progression, then biosecurity protocols have to go in a geometric progression. We have to invest gigantic amount of funding in order to have antidotes for biosecurity measures, because those are the wars which will be fought again. It's just a matter of time. COVID is just one example, but then this is just the beginning of a very new kind of warfare, which is far more sophisticated from what we have seen in 13th century, because you do not need to have that huge cadavers or something to do this warfare. And that brings us to the point, what are we securing? This is what we are securing, and that's what we meant by biosecurity. And when we talk about the biosecurity landscape of human health and pandemics, we talk about some of the things: accidental laboratory leak, accidental spillage, and biological agent or related informations, theft, sabotage, and weaponization of biological agent, gain of function research and dual use of research of concern, foreign invasive species, and agroterrorism. As I mentioned, that you can introduce different kind of weeds and different kind of crops which could destroy the native crop, genetically modified crops, weeds, and field testing, deliberately release infections and toxins for biological agent, and they are all linked to food security. And if you could destroy the food security of a system, then you do not need to wage a war. The economy will falter in no time. Animal biosecurity, import, quarantine, and breeding and diseases. Synthetic infection agent are built using freely available genomic knowledge. So this is, I was telling you, the whole support from the internet you are going to get, because most of these informations are available in the internet, and readily available building blocks, pest, vector-borne diseases, genetically modified, intentionally released insect vectors, and zoonotic spillover of diseases. So this is the landscape which is the basis of investing time and energy to develop next generation of biosensors, because you have to have an antidote unless it is a completely new thing. But then on the hindsight, you can actually develop antidotes for diseases which does not even exist. So we are entering into a very different kind of data mining and simulation, which many of these things will be run through long haul simulation studies, how we can develop different kind of antidote molecules, different kind of antibodies, different kind of vaccines. Most importantly, we have to have the detection systems in place, DNA based sensors, RNA based sensors, protein based sensors, chemical sensors, all sorts of cell based sensors, are very rapid, fast paced sensors. And after next 3 classes, well, I'll be talking about the Ebola, filovirus, insulin, anthrax. We will talk about how we can design these different kind of sensors. And what I will do, as I will walk through, I will again bring up those instances what I could not cover while talking about the different kind of bioterrorist agent which have been used, because that will kind of help you to keep a track of what all areas, if you are interested in this kind of research, what all areas you really have to understand very clearly. Because end of the day, remember when I was telling you about allelochemicals, I told you, the end of the day it will be a chemical. Either the microbe produce a specific kind of chemical which infect your system, or the microbe itself is a agent which has to be nullified, or some other chemical which is infecting the system. Like say for example, one of the historical perspective is that in one of the instance, Napoleon Bonaparte flooded a region to increase the instances of mosquito and to have malaria infection, which is a very innovative thing for the time of Napoleon Bonaparte, but he did it actually. History is there to tell you. So it seems understanding biology, understanding biosecurity, understanding how to design biosensors and how to engineer them is in whole domain is an extremely challenging and dedicated front for study and research. So, in next several weeks to come, we will invest our energy to understand some of these basics, and those of you who are interested, be rest assured, this is one area which, irrespective of anything, will keep growing, because by this time you must have realized if a threat which has started in the 13th century and maybe before that, because we do not have any historical evidences, will continue till the fag end of the civilization. So, I will close in here, and in the next class we will talk about filovirus, which is Ebola, Marburg, and what are the sensors would have, and anthrax, then insulin, and that way we will get a little bit more flair of the different kind of sensors, the need for the sensors and the kind of sensors which are available. Thank you.